Okay, so welcome to this next video in which we are discussing the GQ pathway and the palmitoylation of IP3 receptors. Okay, right. We are just in the process now of finishing our discussion of the GQ pathway, and I explained in length at the end of the previous video why we mustn't stop at the release of calcium from the ER. We need to go further because the release of calcium from the ER into the cytoplasm causes the depletion of calcium stores within the ER, and this is going to activate the store-operated calcium entry pathway, which is going to bring calcium into certain microdomains within the cell, which will activate pathways which calcium that has been released from the ER through the IP3 receptors will not activate. Okay, so let's have our ER here. Okay, so this is the ER. And let's say that calcium within the ER has gone down. Basically, there is a protein in the membrane of the ER, okay, which is called STIM1, okay, so STIM1, which stands for stromal interaction molecule 1. So this is stromal, so the ST is for stromal, then the I is for interaction, and then the M is for, interaction, I can save that, interaction, and then the M is for molecule 1. So this is the stromal interaction molecule 1, or STIM1. And basically what happens is that when the calcium goes down in the lumen of the ER, STIM1 proteins start to stick together. So usually they're on their own in the membrane of the ER. So you'll have more than one, of course, but they're not aggregated together. However, when calcium goes down in the ER, what starts to happen is these things start to aggregate together, so you get STIM1 aggregates being formed. And these STIM1 aggregates can now interact with proteins in the plasma membrane. Okay, so to understand this, you need to understand that the ER is actually quite closely opposed to the plasma membrane. So here's the ER membrane, and then here is the plasma membrane. And basically, these STIM1 aggregates can now activate certain channels within the plasma membrane. So we're in one of these special store-operated calcium entry microdomains now. Let me get my colours to make this simpler. Okay, so remember, here are these walls that are separating off this separate microdomain from the rest of the cytoplasm of the cell. Okay, now here we have our STIM1 aggregate here, so the stromal interaction molecule 1 has aggregated together to form this nice aggregate, and it's going to activate a channel within the plasma membrane, basically. And this channel is what's known as the calcium release activated calcium channel. Okay, so we'll draw it here. So I'll just draw it as blob for now, but we'll have a look at its structure in more detail in a moment. So this is called the calcium release activated calcium channel. So the calcium release activated calcium channel, and if you think about it, it is the calcium release activated calcium channel, because it's basically going to be activated when the calcium has been released from the ER, which results in calcium levels in the ER going down, and therefore STIM1 aggregating. Okay, and for short, the calcium release activated calcium channel is abbreviated to the CRAC channel, which is a rather unfortunate name, but there you are. So C for calcium, R for release, A for activated, and then C for C for calcium again, and then it's the CRAC channel. Okay, right. Let's now have a look at the structure of this CRAC channel in a bit more detail. Okay, so let's pull it out here again. Okay, so I'm going to draw a big picture of it within the plasma membrane. Okay, and now this side of the membrane, the upper half, now represents the extracellular fluid, and for short I'll abbreviate extracellular fluid to ECF. Okay, and this side represents the cytoplasmic side, so this is the cytosol down here. Okay, and you have a little pore going through the middle. Now basically these crack channels, these calcium release activated calcium channels, they are tetramers. They're made up of 4 separate proteins, just like the IP3 receptors were. Okay, and if we now pull out one of these subunits that makes up the crack channel and have a look at its membrane spanning topology, okay, so we'll look at one of the proteins that makes up a quarter of the calcium release activated calcium channel, then basically its membrane spanning topology looks like this. They have 4 membrane spanning alpha helices. Okay, so here is the amino terminus, and here is the carboxylic acid terminus, and between the amino and the carboxylic acid terminus, the protein or polypeptide spans the membrane 4 separate times. Okay, now what are these proteins called? Well, they are called aura I proteins. Okay, so the proteins which you use to make up the calcium release activated calcium channels are called aura I proteins. Now, in the human genome, there are again 3 separate genes for aura I proteins. So there are aura I1 proteins, aura I2 proteins, aura I3 proteins. So I'll just write aura I1 to 3. Okay, however, thankfully for us, the only one that seems to actually be used is aura I1. Okay, and the only calcium release activated calcium channels that you seem to find are aura I1 homotetramers. Okay, so basically this means that you go to the aura I1 gene, you transcribe it 4 times to make 4 pieces of mRNA, you send those 4 pieces of mRNA to the ribosome, and of course this is a little bit over the top because mRNAs can be read multiple times, but just to make the concept utterly clear, you send these 4 mRNAs to the ribosomes, you make 4 copies of the aura I1 protein, and you stick them all together to make an aura I1 homotetramer, and this makes a functional calcium release activated calcium channel. Okay, right. Now, these aura I1 homotetramer calcium release activated calcium channels can then be activated by STIM1 aggregates, and what they will then allow is they'll allow calcium to move from the extracellular fluid here through their pore, okay, which has just opened because of this interaction with the STIM1 aggregate, and the calcium will move into the cytoplasm basically. And of course you will get a net movement of calcium in because the calcium concentration extracellularly is around 1.5 millimolar, whereas the calcium concentration intracellularly is closer to 100 nanomolar. So there is a very powerful concentration gradient, a 15,000-fold concentration gradient favouring the movement of calcium in. In addition, you've got the electrical potential difference across the membrane, which is usually around negative 65 millivolts, okay? So that means that the electrical potential intracellularly is 65 millivolts lower than the electrical potential extracellularly. Calcium is a positively charged ion, so it wants to be where the electrical potential is lower, which in this case is the intracellular compartment. So both the electrical gradient across the membrane and the concentration gradient are both favouring the movement of calcium into the cytoplasm. So what indeed will happen is calcium will move into this little microdomain here. And now what will happen is you've then got a special pump here, which I'm going to highlight in blue, known as the Sarcos/endoplasmic reticulum calcium ATPase. So it's now here in blue. So this is called the Sarcos/endoplasmic, okay, reticulum calcium ATPase, and for short, the Sarcos/endoplasmic reticulum calcium ATPase is abbreviated to the Sarca. Okay, so the S is for Sarco, the E is for endoplasmic, okay, the R is then for reticulum, the C is for calcium, and the A is for ATPase, so the Sarca. Right, so you have these Sarca proteins in the endoplasmic reticulum membrane. Now, what do these Sarca proteins do? Well, basically, they're going to return calcium ions into the lumen of the endoplasmic reticulum. So here is our Sarcos pump. Let's say that this side is the cytoplasm, so this is the side facing into this store-operated calcium entry microdomain, and this is the side facing the lumen of the endoplasmic reticulum. Okay, what's now going to happen is that basically Sarcos is going to pump 2 calcium ions into the lumen of the endoplasmic reticulum. So basically the calcium is coming in through the calcium release activated calcium channel, which is one of these aura I1 homotetramers. It's then being pumped by the Sarcos pumps in the ER membrane into the ER lumen, and in exchange for the 2 calciums that are being moved in, you move 3 protons out, basically. Now, it doesn't just do this on its own accord. Basically, it also needs ATP to be hydrolyzed, so this is an active transport process. So ATP, adenosine triphosphate, will be hydrolyzed to adenosine diphosphate plus an inorganic phosphate molecule that will be cleaved off, and that will release the energy which is used to move these 2 calcium ions in whilst these 3 protons are moved out. Okay, so that is the store-operated calcium entry pathway, and the important thing about this is not just that it allows you to restore the calcium concentration in the ER lumen, but that it also temporarily brings calcium into this microdomain here. And there are all sorts of proteins in here that can be activated by that calcium that's coming in through store-operated calcium entry pathways that are not present in the cytoplasm outside of this microdomain. So certain pathways which are actually, in some cases, extremely important will only be activated by the calcium that is coming in through store-operated calcium entry, and will not be activated by the calcium that was originally released from the ER. Okay, so that now summarizes the GQ pathway. So, let's now turn our attention to what this whole video was about, which was palmitoylation of the IP3 receptors and the role that this plays in stabilizing the IP3 receptors. Okay, right. So, let's talk firstly about palmitoylation and what actually happens in palmitoylation. Okay, so palmitoylation means adding palmitic acid molecules or palmitoyl groups onto proteins. So let me start by showing you the structure of palmitic acid. Okay, so palmitic acid is the old biochemist's name for a molecule that is now also called hexadecanoic acid. Okay, and again, although hexadecanoic acid maybe is a scarier name, it's useful because it tells you exactly what this molecule actually is. This tells you that it's a 16-carbon carboxylic acid that is fully saturated, basically. So, let's start off by drawing the carboxylic acid group here. So here's a carbon with the oxygen double bound to it and an alcohol group coming off. And now we need another 15 carbons. So remember, it's a 16-carbon carboxylic acid. We've got our first one, so we need the other 15. So that means that 14 of these are going to be in the middle as methylene groups. So here is a methylene group. We need to repeat that 14 times. And then right at the end, we'll then have a methyl group sticking off. Okay. So this now is the structure of palmitic acid or hexadecanoic acid. Okay, now, you are not going to use pure palmitic acid to stick onto the sides of proteins. Instead, you are going to use a molecule known as palmitoyl coenzyme A. Okay, and for short, palmitoyl coenzyme A is abbreviated to palmitoyl CoA. Okay, so CoA is the abbreviation for coenzyme A, and this is the same molecule that is so important in the respiratory reactions. And the link between palmitic acid and coenzyme A is going to be pretty much exactly the same link as you have between acetic acid and coenzyme A in acetyl coenzyme A. Okay, so we won't draw the full structure of a coenzyme A molecule. Instead, we'll write coenzyme A and put a box around it. That will do. And the only important functional group of coenzyme A that we need to, actually talk about is this thiol group. Okay, so this is a thiol group. And I would encourage you to think of a thiol group as being analogous in every way possible to an alcohol group. Okay, so sulfur atoms are in the same group of the periodic table as oxygen. They're just underneath oxygen in the periodic table. Okay, so they have very similar chemical properties to oxygen. Okay, so if you imagine replacing this with an oxygen, that would then be an alcohol group. Okay, so thiol groups are actually very similar to alcohol groups. Now, if you imagine for a moment that this actually was an alcohol group, you could link coenzyme A to palmitic acid very easily. You could take the hydrogen off the oxygen of the alcohol group, take the alcohol group off this carboxylic acid group, like so, okay, and combine those two together to make water, okay, and this will be called a condensation reaction because it produces water. You could then bind this carbon atom to this sulfur atom, to this oxygen atom. We're imagining it's an oxygen atom, and that would create a nice link between the palmitic acid molecule and the coenzyme A, and this link would be called an ester link. Now, basically, you can perform pretty much exactly the same reaction for a thiol group as you can for an alcohol group. So thiol groups can react with carboxylic acid groups to create a link that's analogous to ester links. Now, because it involves a thiol group, not an alcohol group, this link is not called an ester link, but it's called something very similar. It's called a thioester link. Okay, so you can link carboxylic acid groups onto thiol groups, and that's what we're going to do. We're going to link the palmitic acid molecule with the thiol group of the coenzyme A molecule to create this molecule that's called palmitoyl coenzyme A. Okay, and it is this palmitoyl coenzyme A that we are going to use to add on to proteins. Okay, so the main type of palmitoylation that occurs is what is known as S-palmitoylation. Okay, and this is a post-translational modification that you can make to proteins. So basically what happens in S-palmitoylation is that you add palmitoyl groups onto cysteine residues within proteins, and this is very important. It's only cysteine residues that you can add palmitoyl groups onto. Now, of course, proteins can have many, many different cysteine residues. Are you going to add palmitoyl groups onto every single one? Well, of course not. And what is going to happen is that certain cysteine residues within the protein are going to end up being palmitoylated. And this is going to help the protein to attach to membranes, basically. It's going to stabilize it within within phospholipid bilayers, because the palmitoyl group is very hydrophobic. Okay, so it will interact very favorably with the other hydrophobic molecules within phospholipid bilayers. Okay, and that can help to stabilize proteins which are within membranes in those membranes. Okay, and we're going to see that this happens to IP3 receptors, or at least it's a putative claim, and I will give you the reference that claims this, but it's probably true. Okay, and it's certainly an interesting model, so we'll discuss the model basically. All science is just models, remember. Everything, nothing is proven. Everything's just models. I mean, there are good models that we're pretty sure are right, but they're never proven. Okay, so this is a model. Right, so we'll start off with a discussion of S-palmitoylation, and this is a very, very good model. This isn't putative at all. Okay, so what's going to happen is that you're going to add palmitoyl groups onto certain cysteine residues within the protein. Okay, so let's draw a cysteine residue. Let's start off our discussion by showing a cysteine residue here. So here is the core amino acid structure. So we have the amino group, the alpha carbon, the carboxylic acid group, and then the R group of a cysteine residue consists of a methylene group with a thiol group sticking off it. So here is the thiol group. Okay, now what you are going to do is you're going to bring in your palmitoyl coenzyme A molecule. So let me show it here. So here's the carboxylic acid group that's linked via this thioester link to coenzyme A, which is here. Okay, and then the R group of the palmitic acid molecule is 14 methylene groups. Okay, so I'm doing this trick of drawing a single methylene group and then putting brackets around it and then subscripting it 14, and then finally a methyl group on the end. Okay, so we're bringing in our palmitoyl-CoA enzyme, molecule, and what we're going to do is we're basically going to cleave the bond between this carbon and this sulfur. Now remember, covalent bonds consist of 2 electrons, basically, one from each of the 2 members of the bond. So there will be 1 electron from the sulfur atom and 1 from the carbon. Okay, imagine giving the electron from the sulfur back to the sulfur and giving the electron from the carbon back to the carbon. Okay, now break this covalent bond between this sulfur and this hydrogen of the thiol group of the cysteine residue of the protein. Okay, and again, imagine giving 1 of the electrons back to the hydrogen and 1 back to the sulfur atom. Now, bind this hydrogen atom, which has a free electron, to this sulfur atom, which has a free electron, to regenerate coenzyme A, basically. Okay, with its original thiol group, fully regenerated. And then bind this sulfur atom, which has a free electron, to this carbon, which has a free electron over here. And effectively you have now transferred the palmitoyl group off the thiol group of the coenzyme A and onto the thiol group of the cysteine residue. And again, it's linked to this cysteine residue by a thioester link, basically. Okay, so basically this reaction requires an enzyme to catalyze it. It doesn't just occur on its own. But once it has occurred, you have palmitoyl groups stuck onto the sides of proteins. Now, the claim, okay, and this claim is made by a paper that was published in October 2014, which I have here. Okay, and if you want to go and read the paper, it's here. So this is the title that you can Google. So, stable expression and function of the inositol 1,4,5-trisphosphate receptor requires palmitoylation by a DHHC6 selenoprotein K complex. Okay, so if you want to go and read that paper, you can. And that's a primary research paper, so it has all of the experiments they did to try and elucidate this. Okay, and in upcoming videos I'm going to describe some of the experiments they did with a full discussion of the experimental method, and that's the main aim of it, to understand the experimental method. And we'll subsequently look at this same problem and try and understand why each of the experiments helps them to show that. Okay, so back to S-palmitoylation then, before we get on to that paper. And that's why I say that this is a putative claim. This has only been claimed since October 2014, so it's new stuff, basically. Okay, but it's a nice paper and I would recommend you read it. Alright, so S-palmitoylation does not occur on its own, basically. It requires enzymes to catalyze it. Now, there are 23 enzymes that are capable of catalyzing this, and they are all in the family of DHHC enzymes, okay? And all of these DHHC enzymes are palmitoyl transferase enzymes, so they're going to transfer palmitoyl groups from coenzyme A molecules onto cysteine residues within proteins. Okay, right. In the next video, we will look at a specific example of the DHHC enzyme, which is the one that is claimed to palmitoylate cysteine residues within IP3 receptor subunits, which is the DHHC6 enzyme, and the claim that it associates with another protein, which is called selenoprotein K, to form a functional complex, which will then palmitoylate receptor subunits of the IP3 receptor. Okay, so we'll do that in the next video.